Hi, I am Dr. Sabin. Today I am going to talk about type 2 hypersensitivity reaction. The ultimate result in type 2 hypersensitivity reaction is lysis of the target cell or cytotoxicity. And the cytotoxicity occurs in response to IgG, IgM, or IgE, which are synthesized against endogenous or exogenous antigens. Because there is a role of antibodies in the pathogenesis of type 2 hypersensitivity reaction. It is also known as antibody-mediated cytotoxic hypersensitivity or simply antibody-mediated hypersensitivity. Now let's study endogenous antigens and exogenous antigens. Endogenous antigens are usually self-antigens and they are more commonly implicated in the pathogenesis of type 2 hypersensitivity reaction. Exogenous antigens are environmental antigens. They are present on cell surface. Endogenous antigens can be present on cell surface or sometimes in extracellular matrix. Endogenous antigens can be on cells like red blood cells, cell receptors like TSH receptor, acetylcholine receptor can act as endogenous antigens. Sometimes antigen can be found in extracellular matrix. In Goodpasture disease, endogenous antigen is found in type 4 collagen of glomerular basement membrane and alveolar basement membrane. Several types of drugs, blood group antigens, and microbes can serve as exogenous antigens. Now, let's have a look at the sequential steps in type 2 hypersensitivity reaction. Antibodies, which are usually IgG, IgM, or IgE, are produced against an antigen. The FAB part of antibody, this is the FAB part of antibody, binds to the antigen on target cell. This is target cell. For example, it can be red blood cell or platelet. This is the antigen on the target cell. So FAB portion of antibody binds with the antigen on a target cell. Effector cell, which can be macrophage, eosinophil, basophil, neutrophil, or a mast cell, binds with the Fc portion of the antibody via its Fc receptor. This is the Fc portion of antibody. In this diagram, this is our target cell. This is antigen on the surface of the target cell. Antibody is produced against the antigen. FAB portion of antibody binds with the antigen. This is the Fc portion of antibody. This portion binds with the Fc receptor of an effector cell. An effector cell can be a neutrophil, an eosinophil, a macrophage, a basophil, or a mast cell. After an effector cell binds with the antigen and antibody complex on the surface of a target cell, the target cell is destroyed by one of the 5 mechanisms. These mechanisms are antibody-dependent cellular phagocytosis, complement-dependent cytotoxicity, ANCA-mediated cytotoxicity, antibody-dependent cell-mediated cytotoxicity, and the 5th one, antibody-mediated cellular dysfunction. In the following slides, I will be describing each of these mechanisms in detail. In this slide, I will describe the first mechanism of type 2 hypersensitivity reaction, that is antibody-dependent cellular phagocytosis, ADCP. This mechanism occurs primarily when the antibody formed against an antigen is of IgG class. The antibody IgG binds with the antigen on the surface of a target cell. This process is known as opsonization. A phagocytic cell binds with the Fc portion of antibody through its Fc receptor, and the target cell with antigen is engulfed by the phagocytic cell. Macrophages, neutrophils, and eosinophils are phagocytic cells. Immune thrombocytopenic purpura, ATP, is a good example of antibody-dependent cellular phagocytosis. It is an autoimmune disease. Antigens are present on the surface of platelets. The antibodies are of IgG class. Opsonized platelets are phagocytosed by macrophages in the spleen. In this way, there is extravascular destruction of platelets in the spleen in immune thrombocytopenic purpura. Antibody IgG is a good opsonin. When this antibody binds with an antigen, efficiency of a phagocytic cell increases. But in a situation when an antigen is present on a large surface, a phagocytic cell cannot engulf the antigen despite the presence of opsonization by IgG. In that situation, phagocytic cell releases cytolytic enzymes to kill the antigen, but tissue injury occurs. Such a mechanism is called frustrated phagocytosis. Goodpasture disease is a good example of frustrated phagocytosis. In this disease, antibodies IgG are synthesized against an antigen on type 4 collagen of glomerular basement membrane and alveolar basement membrane. IgG antibodies bind with the antigen and immune complex is formed. The immune complex activates classical pathway of complement system incompletely. C5a complement fragments are generated, which are chemotactic for neutrophils. Neutrophils bind with the Fc portion of IgG and are activated. Activated neutrophils release cytolytic enzymes and reactive oxygen species. The molecules injure tissue. Tissue injury caused by the cytolytic enzymes and reactive oxygen species in glomeruli leads to disease called anti-GBM nephritis. When alveolar basement membrane is also affected, the disease is called Goodpasture disease. Lysis of target cells by membrane attack complex of complement system or complement-dependent cytotoxicity is the second mechanism of type 2 hypersensitivity reaction. This mechanism is seen primarily when antibodies against the antigens are of IgM class. Acute hemolytic transfusion reaction due to ABO incompatibility is an example where the target cells are destroyed by activated complement system. Now let's look at the sequential steps of complement-dependent cytotoxicity, taking acute hemolytic transfusion reaction as an example. When blood of group A person is given to a person who has a different blood group, there will be acute hemolytic transfusion reaction. The recipient already has preformed IgM antibodies against the donor red blood cell antigen. So the preformed antibodies of the recipient will bind with the antigen present on the surface of the donor red blood cell, and this is immune complex formation. This illustration shows the complete activation of complement system. In acute hemolytic transfusion reaction, IgM antibody binds with the antigen on the surface of donor red blood cell. Membrane attack complex is formed on the surface of the red blood cell. 18 molecules of C9 are polymerized into a tube-like structure. The tube is inserted in the plasma membrane of the red blood cell. Water enters the red blood cell through this tube, and ultimately there is osmotic lysis of the donor red blood cells. Antibody-dependent cell-mediated cytotoxicity (ADCC) is the third mechanism of type 2 hypersensitivity reaction. Macrophages, neutrophils, eosinophils, and mast cells are capable of causing tissue injury through ADCC. Physiologically, ANCA cells kill tumor cells, self-reactive cells, and virus-infected cells through this mechanism. Lafleur's syndrome caused by Ascaris lumbricoides is an example of ADCC. Adult worms of Ascaris live in the small intestine, but the larva form has to pass through the lungs during its life cycle. During its passage through the lungs, the larva induces inflammation there. The inflammation kills the larva, but it also causes tissue injury in the lungs. The tissue damage caused by the larva in the lungs is called Lafleur's syndrome. Now let's study the steps of antibody-dependent cellular cytotoxicity (ADCC), taking Lafleur's syndrome as an example. IgE antibodies are synthesized against the Ascaris larva antigens. The Fab portion of antibody binds with the antigen on the surface of the Ascaris larva. Fc portion of antibody binds with the Fc receptor on the surface of the eosinophils. This is larva of Ascaris lumbricoides. These red dots represent antigens on the surface of the larva. IgE antibodies are synthesized against these antigens. Eosinophils bind with the Fc portion of antibody through the Fc receptor and they are activated. Activated eosinophils release cytolytic enzymes to kill the larva. In addition, the tissue is also injured. The cytolytic enzymes released by eosinophils are eosinophil cationic protein, eosinophil peroxidase, and eosinophil-derived neurotoxin. ANCA-mediated tissue injury is the fourth mechanism of type 2 hypersensitivity reaction. ANCA stands for anti-neutrophilic cytoplasmic antibody. Proteinase 3 and MPO are found in the primary granules in the cytoplasm of neutrophils. Because of certain drugs and infections, these enzymes are translocated to the surface of the neutrophil. On the surface of the neutrophils, these enzymes act as antigens and they are exposed to the immune system. Immune system synthesizes antibodies of IgG class against these antigens. When these antibodies bind with the antigens on the surface of the neutrophils are activated. Activated neutrophils release cytolytic enzymes and reactive oxygen species with the result of vasculitis. CNCA or antiproteinase 3 ANCA causes Wegener's granulomatosis. The new name and the preferred name for Wegener's granulomatosis is granulomatosis with polyangiitis. PNCA or anti-MPO ANCA causes microscopic polyangiitis and some cases of drug-induced vasculitis. Antibody-mediated cellular dysfunction is the fifth mechanism of type 2 hypersensitivity reaction. In this mechanism, antibodies are synthesized against cell receptors. When the antibody binds with the target cell receptor, function of a cell may be increased or decreased. Graves' disease and Myasthenia Gravis are caused by this mechanism of type 2 hypersensitivity reaction. In this illustration, this is thyroid follicle. TSH receptor is present on the surface of thyroid follicular epithelial cells. In Graves' disease, IgG antibodies are synthesized against TSH receptor. These antibodies stimulate the follicular epithelial cells. That's why these antibodies are called thyroid stimulating immunoglobulin. In Graves' disease, because of the stimulation of follicular epithelial cells by thyroid stimulating immunoglobulin, hypersecretion of T3 and T4 occurs. A patient with Graves' disease presents with signs and symptoms of hyperthyroidism. Myasthenia gravis is an autoimmune disease caused by type 2 hypersensitivity reaction through antibody-mediated cellular dysfunction. Antibodies are produced against nicotinic acetylcholine receptor in the postsynaptic membrane of neuromuscular junction. Antibodies are usually of IgG class. Sometimes IgM and IgA antibodies are also synthesized in lower titer. Antibodies bind with acetylcholine receptor and block the action of acetylcholine. Complement system is also activated. Acetylcholine receptors are destroyed and postsynaptic membrane is damaged. Neuromuscular transmission of nerve impulse cannot occur. Therefore, patients present with progressive skeletal muscle weakness.